Am I on? Yes? Okay. Welcome, everybody, to part 2 of Hallmarks of Aging. I'm told by Steve that we're on live. I guess we are. No? Yes? Yes. Okay. So welcome to... So he's making all kinds of faces. I guess this is the Halloween show right now.
He does this every month. He does this, folks. He says, "Are we on? Are we on?" I've already said like, yeah, we're on. So about 4 times.
It's very ambiguous gestures you make. Anyway, welcome to part 2 of Hallmarks of Aging. I'm Oliver Medvedik. I'm broadcasting from Cooper Union in the city of New York at the Cambar Biomedical Engineering Laboratory, and I'll let everybody else introduce themselves, starting with Steve. We have a panel of folks joining us besides Steve.
Okay, well, I'm Steve, and I'm part of Leaf, and I'm always here, so you know I'm a bit like the janitor. Hello, folks.
Mm-hmm.
Happy Halloween. Who's next?
Joel.
Hoorah. My name is Joel. I'm from Reno, Nevada. I've been sick most of my life, and now I've become a consultant for sick people, sick families, mostly veterans and people burnt out on the system. Okay. And I've become a researcher to back up all that to the latest medical field and advancements. Nice to meet everybody.
Cool. Sven.
I'm Sven. I'm a student in biochemistry and biotechnology at Ghent University, and I'm also the co-founder of HEALS, which is a European non-profit organization that focuses on aging and promoting aging research.
Nice.
Cool. Very cool. Laura. Hello, Laura.
Hello. Hi, I'm Laura.
I'm also the co-founder.
And I'm a pathologist by trade and also a volunteer for Leaf.
Where are you calling from? Is this from the UK?
England, yeah. Yes, as you can probably tell by the accent.
Yeah, well, you know, people are global these days.
Mm-hmm.
And are you introducing yourself? Asset Gebnut? No? Lurking in the background?
Mm-hmm.
Yeah. Awesome. Well, we may or may not have Bill. It's hard to tell, but let's proceed with the journal, shall we? So we have a review paper. It's the Hallmarks of Aging, and let me just share my screen with all of you. Okay, so it's the Lopez Autin et al. Paper, Hallmarks of Aging. We went over a number of hallmarks, or roughly half of them last time. So you should see this wheel that illustrates these hallmarks. And this is basically the attempts of these authors to essentially bin or categorize all the various things that go wrong in aging, not necessarily chronologically. Some of these overlap, some of these happen at the same time, and some of them lead to other consequences. So last week we talked about genomic instability, basically what can go wrong with DNA and the different ways that DNA can break down through a variety of insults that happen, intrinsic and extrinsic. We talked about telomere attrition. So that's also related to genomic instability. So telomeres are basically the caps at the ends of chromosomes, linear chromosomes that basically degrade over time as the cell replicates, unless the cell has a means of replacing telomeres, usually through an enzyme, and that enzyme being telomerase. Then we discussed epigenetic alterations. So one hallmark of aging that happens is not only do you have genomic instability, but you have epigenetic modifications happen that change over time. So methylation status changes, acetylation status changes, and it changes in a variety of different locations, not evenly throughout the genome. And that may influence genomic instability. And last, we talked about loss of proteostasis. So proteins are, as you all know, complex three-dimensional nanomachines of the cell, and they need to maintain their complex structure to perform all their roles. So when that doesn't happen, when they misfold, there's a number of problems that can happen. Loss of homeostasis. So there's a number of systems in the cell to help get rid of problematic proteins, also refold proteins via the chaperones, the heat shock proteins, for example. Okay, so those are the 4 things I believe we discussed last time. And we discussed some of I don't know if we got into some of the potential treatments, but we'll certainly talk about that again this week. So next up, we're going to talk about deregulated nutrient sensing. And this is so I'm going to scroll through. So this is the genomic and epigenomic alterations section. Scroll. Okay, so now we have this rather complex looking chart, which is actually probably overly simplified, if you want to believe that. And basically, kind of the nexus point of all of this, really the conserved pathway that all organisms, I guess, that have been studied, model organisms, is this IIS pathway, which is insulin and insulin-like signaling pathway. So it's called insulin and insulin-like signaling pathway because downstream of the receptors, the pathways are the same or similar. And these pathways are affected by one of the classic means by which we extend lifespan, which is dietary restriction. And there's a certain enzyme or a kinase that forms 2 complexes, mTOR, which is sort of at the nexus point of this nutrient sensing and signaling pathways. And this has been really a target of great interest. It stands for target of rapamycin, because it can influence a lot of downstream effects that can directly impinge on aging. So basically what this system does is it signals if there's not enough nutrients, the metabolic system can go into essentially high gear. So if you have glucose coming in, it can be utilized much more readily. There's a lot of parts here that we can look at independently, but is there anything anybody wants to interject in this pathway? I know, Steve, you had a few things you mentioned last time about.
Well, just to say that it's more than just caloric restriction, as some people insist on saying whenever this topic is mentioned. I'm not saying any names, but you know who you are. And yeah, the fact of the matter is there are 4 distinct pathways involved in this. You've got 2 catabolic and 2 anabolic, and they make up the sum of what most people understand as being metabolism. So it's not just a case of, oh, it's just caloric restriction at all. So it really is quite complicated. Isn't it really, Oliver? I mean, that diagram is delightfully simple.
Yeah.
But yeah.
Can I ask a question?
Mm-hmm.
Is this purely based on glycolysis, or is this also involving sort of ethanol and ketone-based sort of metabolic pathways?
Well, mTOR senses proteins or amino acids, right? So it's basically plugged in to not just glucose or carbohydrates, but also
so it's
proteins.
anything that's broken down. Okay, cool.
So that's why it's such a central mechanism. And here, if we look at some of the pathways that some of the key players here, some of you might recognize some of these guys here. SIRT1, which is a histone deacetylase, and also deacetylates a number of other target proteins. And SIRT1 has been certainly implicated in aging or reduction of aging. And this is all color-coded, by the way. So if we scroll down here, molecules that favor aging are shown in orange. So all of these things in orange, if you knock them down, they should lead to an extension of lifespan, or if you not knock them down fully, but repress them. And everything in green, essentially, are things that if they're upregulated or they're somehow enhanced, or potentially by using small molecules, aging is diminished, or the rate of aging is diminished. So I don't know what else to say about this pathway. Anybody have any questions?
Well, there's one thing to say about the pathway, the IIS pathway. There's a seeming paradox with caloric restriction, which caloric restriction reduces the levels of growth hormone and IGF-1, and also it falls with when you get older, those levels fall as well. And people couldn't understand there was a seeming paradox because they couldn't understand that CR, which is generally regarded as beneficial to health, but it did the same thing as aging, which obviously isn't beneficial to health. And they thought, well, that's a paradox. But it's not really. And it's explained actually quite well in Hallmarks. Well, I personally thought it was. What it is actually the body putting the brakes on metabolism as you get older. So it pushes down IGF and it also reduces, those levels. Much the same way as CR. So it's putting the brakes on to try and stop you basically from dying. So there's no paradox there at all, which I thought was quite interesting because they couldn't work out. They couldn't work it out for years. So imagine if your body didn't put the brakes on and you churned out as much IGF as you did when you were younger. Presumably, you would die significantly earlier than you would. So it's interesting how the body tries to fight and survive at any cost.
Yes. So the supposed paradoxes are really compensatory mechanisms. So they're not really, so these things being deregulated is somewhat of a survival, survival mechanism. Certainly if you reduce metabolism, this is beneficial with regards to suppressing cancer or tumors. That's another aspect of some of these mechanisms, like cellular senescence, for example, is considered to be a mechanism by which tumors may be shut down. But too much of cellular senescence will have a negative effect. So one thing to keep in mind with all of these pathways is that there is an optimal level of activity here, and you can't shut them down fully, and you can't crank them up fully, right? There has to be a certain optimal level of activity taking place. And that optimal level is what deregulates over time. And certainly the deregulation could be compensatory, meaning it could, suppress cancer. But if it goes too far, it may start suppressing other things like stem cell renewal. So there is this balance that needs to be maintained. Is that, you think, a decent summary, Steve?
Yeah, it's interesting, and it is really the core mechanism behind metabolism. But I mean, it is further down in the list of hierarchies of damage anyway. So it's more of a sort of secondary aging damage, which is provoked by the ones that we've gone over initially, the first four, because that's something that Hallmarks does that other things like SENS don't. They actually attempt to put a hierarchy, and this particular damage is the first of the secondary damages. In effect, DNA and the other ones above this are kicking down and affecting it, and that's that's bad news. But the question is really, if we fix the primaries, will that help stabilize this to a certain extent? It possibly will. Let's find out.
Yes, certainly. Let's find out. So, right underneath metabolic, well, coupled to metabolic alterations, and right underneath figure 4A is figure 4B, which is mitochondrial dysfunction. And that's a pretty big one, because mitochondria basically provide the bulk of your energy, your ATP, and mitochondria have been looked at for quite a long time with regards to aging, and particularly through the generation of this ROS that's here, which is shown in this pathway, which is a reactive oxygen species. And for a long time, it's been thought that eating lots of antioxidants is the way to go, because you want to get rid of all your reactive oxygen species. It turns out that they're necessary perhaps for signaling and for other roles, and that and there's been a lot of conflicting models out there where feeding antioxidants doesn't really have much of an effect on certain systems, I believe, like in mouse models. But there's a number of things that can still go wrong with mitochondria. Basically, since mitochondria are very metabolically active, you can certainly have protein misfolding taking place, mitochondrial DNA mutations, and all of these damages would have a really dramatic effect on the cell. For example, not just shutting down energy, but certain leaky mitochondria are also the nexus point for apoptosis, since a number of proteins that are associated with mitochondria regulate apoptosis. So if you have a deregulation of mitochondria, you not only have a churning out of maybe excess reactive oxygen species, which can go on and damage things not only in mitochondria but outside of mitochondria. You can have defective apoptotic responses, perhaps enhanced cell death, and also much lower energy levels, much lower ATP levels, which will pretty much broadly affect every system across the board in a cell. And the cell does have ways to circumvent this. One system or mechanism that's being studied now is mitophagy, which is basically a variant of autophagy in that defective mitochondria are degraded and gotten rid of. Is there anything on this topic anybody wants to interject or talk about? No?
Well, I don't want to be the only person who's doing the talking.
Okay.
Hang on, I do have. Wait, sorry, I'm just looking at my scribbles. I do have a question about. Yeah, sorry, I can't read my own notes. Uncoupling protein 1, which causes the non-shivering thermogenesis in brown adipose tissue. I understand that upregulation of that has a beneficial sort of longevity effect. Have there been any other studies on the other homologs of the other uncoupling proteins in mitochondria?
I really don't know.
Fair enough.
Does anybody else know?
No clue, really.
There's a lot of catching up we need to do. There's a lot of.
I do know that a few years ago, Jan Huber has tried DNP, dinitrophenol, in C. elegans as an uncoupling reagent. But I actually don't know if the results have been published of that work, and I didn't remember what the results were.
Okay.
They do mention dinitrophenol in the paper here, in the review paper, perhaps as an uncoupling system. I'm just scrolling through here. Okay, so this is on page 1204, the top right-hand top paragraph. Finally, mitochondrial uncoupling, either genetically through the overexpression of the uncoupling protein UCP1, as you mentioned, or by administration of the chemical uncoupler 2,4-dinitrotoluenol, can increase lifespan in flies and mice. So there's a number of citations there. They end at 2010, so there perhaps are additional ones that I'm not aware of.
Does anyone know the exact reason why, other than the fact that it would obviously break down excess fat, which would take, obviously more stress off the system altogether?
No.
Okay.
I don't. Okay. So, what is the next hallmark? Cellular senescence. So, that is yet another huge hallmark that's studied by a number of researchers, including Judith Campisi. Share the screen here. I guess they don't have a picture of cellular senescence, but we're going to be up here. So cellular senescence is essentially cells entering a quiescent state. So it's a non-dividing state that isn't considered normal, although that's debatable. And cells senesce for a number of reasons. The kind of classical reason, or originally, senescence was identified in the early studies of Leonard Hayflick and the Hayflick limit back in the early 1960s, where cells, primary cells from humans, had a limited replicative potential. And that was attributed later to the lack of telomerase. So the cells essentially had telomeres that were shrinking during every replication cycle. And as a result, the cells had mechanisms in place that sensed DNA damage and prevented the cell from replicating further. There are other systems there that can implement senescence that don't involve telomeres. So if there's other damage that occurs, there is this tumor suppressor pathway, this P16 INK4A retinoblastoma pathway, which essentially halts the cell cycle. And in this paper here, they mention that what's striking is that the levels of P16 INK4A, they correlate with the chronological age of essentially all tissues analyzed both in mice and humans. And they say, we are not aware of any other gene or protein whose expression is so robustly correlated with chronological aging across tissues, across species, and with a range of variation that on average is 1 order of magnitude between young and old tissues. So that's why you see, P16, promoter for P16 being used as a, essentially a sensor for senescent cells, for detecting senescent cells, because it's so broadly turned on in senescent cells. The problem with senescent cells is that they don't really make good neighbors. They basically have this phenotype, which is called SASP or SASP, senescence associated secretory phenotype, where they essentially release a number of factors that promote basically inflammation. So, and this leads to a condition known as inflammaging, where you essentially have inflammation go into high gear. You have excess damage occurring from the immune system, and that damages tissues, surrounding tissues. Again, just as we mentioned with just as we mentioned with the previous examples, particularly nutrient sensing and signaling, senescence may have its role. Certainly as cells grow older and they accumulate mutations, you don't want them to keep dividing and potentially turning cancerous. So having them enter some kind of quiescent state may be good, but at some point, if you have too many senescent cells accumulate, tissue homeostasis basically goes out the window. So it's interesting in that we've discussed some papers recently where the clearance of senescent cells had very beneficial effects on the organism. The question is, will this be beneficial if you keep doing it? Because you're basically clearing out senescent cells, but you're not replacing cells. What else can we talk about cellular senescence here?
Well, you asked that question, but in experiments, when they have purged senescent cells, we see an increase in, sorry, weird noise, tissue regeneration. So, less inflammation improves the situation almost certainly.
Well, yeah, it does improve it. The one thing that I guess, maybe a little concerned about is this is kind of leading into the next one, which is stem cell exhaustion, which is the decline in regenerative potential of tissues, right? So that's another characteristic of aging. So I guess one of the classical hallmarks is decrease in hematopoiesis. So basically you have less and less differentiated immune cells that are primed and ready to recognize threats, and that seems to seemingly there's a decrease in stem cells not only in the hematopoietic system but throughout other tissues as well. Yeah, sorry. I was just saying if there's anything additional I could talk about when it comes to stem cell exhaustion. The one thing I made a note to myself of is this term exhaustion, right, which is used a lot. How much is really known about exhaustion? I mean,
Not that much.
That seems like a very important word, right, that's being thrown around.
It depends how you define exhaustion.
Well, you run out of you don't have the capability anymore to maintain homeostasis of a tissue because of a decline in stem cells. And that happens in a number of tissues, not just related to aging, but also, certain pathologies like obviously type 1 diabetes, where you have an exhaustion of cells that produce insulin, your pancreatic beta islet cells. You have exhaustion of your helper T cells when you're infected chronically with HIV, right? Because they're replicating over and over again. They're being killed off, and they're, trying to regenerate the system, and then they're exhausted. How much is really known about that word exhaustion on the molecular level is my question. I mean, is it just a decrease in telomeres? Is it accumulation of damage?
Is it spans over everything? Because is it, like you said, is it the telomeres thing? Is it the fact that they are unable to access the means, the sort of the building blocks anymore to actually put the stem cells back together? It's, are there external factors coming in and,
But why do they exhaust?
Well, even stem cells eventually replicate so many times. Not all stem cells, but there are certain types of stem cells that just run out of steam. It's just because they produce telomerase doesn't mean that they can keep going ad infinitum. And
Why not?
The thing is, stem cell mobility is also limited by its signaling environment. I don't know if you're familiar with the work of Irina Comboy, but they showed that signaling environment, in particular pro-inflammatory cytokines like TGF-beta, TNF alpha, the usual suspects. Once they sort of reach that tipping point, it literally shuts off stem cells. So it forces them to become dormant or quiescent, and they no longer work. So even if they're still functionally able to work, it's a moot point because they don't. They just remain dormant. And some people think there's quite a lot of stem cells that are just sitting there, but they're being literally inhibited by the amounts of, inflammatory cytokines. Mm-hmm. So the question is, and this is why, just to go back to senescent cell clearing, which is a major contributor to inflammation, what's exciting for me is, will removing those senescent cells, thus reducing the amount of systemic inflammation, improve stem cell function? And it should do.
But is that really the main I guess the main reason why you have exhaustion, in that you don't run out of stem cells, you have plenty of stem cells, they're just not doing anything?
Well, there were some experiments in mice in the hematopoietic stem cell compartment, and they did serial transplantations, and they showed that you could keep the stem cells alive for longer than the normal lifespan of mice. So the stem cells kept just rebuilding the entire blood compartment of the new mice in which they were transplanted. So that seems to indicate that the environment is really important and that you are not really running out of stem cells. Though this is one stem cell compartment and it's in one organism, we don't know if this also applies to humans.
Is this so? I've correct me if I'm wrong, but aren't there other reports that suggest that if you do serial transplantation, they peter out after a time period, that you can't keep doing this ad infinitum?
Well, I guess that ad infinitum is probably not possible for everything, but I'm not aware of all the literature, but I know of at least one study where you could do that for longer than the lifespan of mice.
Okay. Yeah, that's something that's always been in the back of my mind, and that's something I want to read up a little more, is just that word exhaustion, which seems to be a very, very broad, hand-wavy term. And what are all the molecular details? Are there different forms of exhaustion? I mean, and I'm talking about in situ. Yeah, clearly you exhaust stem cells if they run out of telomerase and their telomeres shrink and they undergo senescence. Sure. But what about in situ in these niches? Why do you have exhaustion happen in the different organs, the different tissues, right? Why exactly do you run out? Are there different mechanisms, or is there one conserved mechanism that ties them all together? I don't know.
I think some populations of stem cells do genuinely run out. I think it's the hypothalamus. Yeah. There's a certain type of unique stem. I'm sure it's hypothalamus, and they actually run out. Bad things happen when that particular sort of subset of stem cells run out. But they've been experimenting with in situ reprogramming and turning, I think it was astrocytes, and they turn them into this other more rare form of stem cell in situ, so that and it improved the situation. So I think it varies depending on which populations of stem cells you look at. Certainly, like the blood stem cells, HSCs, they do get depleted as you age, and of course, as they get depleted, that also runs your immune system down, because, as you know, the blood stem cells give rise to all the other immune cells in the body. So they do run out.
Mm-hmm. So I'm just reading the paper a little bit here, 1206, their take on stem cell exhaustion. And again, it's complex. They mention a number of things that, correlate with stem cell exhaustion. Increased damage, overall damage that goes up, decrease in telomeres in multiple tissues, and this causes an increase in, P16 INK4A, which inhibits the cell cycle. And then they talk about importance of stem cell quiescence for long-term functionality of stem cells being compellingly demonstrated in Drosophila with intestinal stem cells, in which excessive proliferation leads to exhaustion and premature aging. So this is Rera et al. 2011. I remember another paper, God, I have to pull this out, where they were studying asymmetric damage accumulation in stem cells in Drosophila. And they were looking at intestinal crypt cells. And so they were looking at the stem cells in the intestine, and as they differentiated, to see where damage was being shunted. And as I recall in the paper, most of the damage was being shunted to the differentiated cells, as would make sense. It's a mechanism for getting rid of damage. And yet, seemingly, the functionality of stem cells, even though they have these mechanisms in place to kind of, I guess, get rid of damage, it's still not enough. They still enter, senescence. They still get depleted. So I don't know. I'm still not clear as to what are all the contributions when it comes to the word exhaustion.
Inflammation. From its various sources. It's definitely a major player in stem cell function, ability, and its ability to remain, alive. I mean, bearing in mind that things like telomerase, which stem cells use to renew themselves and keep on trucking, that can be inhibited by inflammatory cytokines as well. So as systemic inflammation rises in the body with age, and you look at the different sources, you've got senescent cells, you've got microbial burden, and microbial burden seems to be potentially the primary source of where inflammation starts, because they took germ-free mice, very recently, and they found that these mice had no age-related cytokines at all, and their macrophages and everything worked properly. At the moment they were mixed with normal mice, they started to get inflammation. And if they were given the microbiome from inside normal mice, and it was injected into these germ-free mice, again, they'd start aging really fast with the same signals. So it suggests that the microbiome might be where the first sort of spark begins, and we know that inflammation damages macrophage function, as Goodcoff and various other people have shown. So then the macrophages, the neutrophils, and all these other things that are clearing up debris stop doing it. Then you get senescent cells accumulating, so you get more inflammation. Cell debris make more inflammation, and it just goes round and round and round. And the problem is, the more inflammatory signals you get, the more it's pushing down and suppressing stem cells, and then you're losing tissue regeneration. Ultimately, it just shuts them all down.
So if that's correct, then taking a particular multipotent stem cell, harvesting it, growing it in a dish, giving it all the right factors it needs, upregulating telomerase, it should be immortal like a cancer cell. You should have propagated indefinitely without any problems, or the population.
Well, for a long time, yeah.
Yeah.
I mean, really, they are self-perpetuating to a certain extent. Yeah. I mean, they get damaged, but they're pretty robust. Irina Conboy, in a paper last year, she was saying that there is really functionally not much difference between an aged stem cell and a young stem cell if given the appropriate signaling. I mean, you can see just by knocking out TGF-beta with ALK5 inhibitors in a 2015 experiment, an amazing sort of transformation in tissue, in muscle, I think it was brain, and was it skin? It's at least 3 tissues, and just knocking out one cytokine, or not knocking it out, reducing it to youthful levels, allowed the stem cells to start coming online again. So a lot of it's going to be inflammation based, but some stem cells do become so damaged over time, you do lose them. Mm-hmm. But it begs the question, how much of a player is inflammation? And all the signs so far are it's a huge one.
Well, over long periods of time, you will always have these types of, random events that lead to one stem cell dying from some, trigger that, damages the cell too much and it cannot repair in time and it dies. And so over long periods of time, even in perfect conditions, you will have loss of stem cells. But, probably we will be able to fix that with just transplanting new stem cells.
And another thing on a related note is, a couple of months ago, they found a new technique for locally reducing systemic inflammation to facilitate transplants of stem cells. What they found was by reducing inflammation at wound sites and then introducing the stem cells from an external source, they found out that firstly, the grafting was better, where it joins the tissue. And secondly, the survival rate was considerably better. And that was just by reducing the local level of inflammation, because not only was it reducing the cytokines, it was also modulating the immune system locally. So like the macrophages, the T cells, particularly macrophages, which were favoring a more aggressive inflammatory sort of profile, which is called a polarization. They went from M1, which is the aggressive type, to M2, which is a soothing, calming, healing type of macrophage or profile. And it was interesting because it then facilitated the engraftment of new stem cells. So inflammation is a huge, huge problem.
Interesting. How does that tie in with, obviously with all the work that's been done with clearing senescent cells, right? Is this, what happens to senescent cells in the tissue when they have they looked at? Well, obviously, if you reduce inflammation, do you get a reversion of many senescent cells? Do you have a decrease in the population?
Quick question.
Yeah.
Has there been studies on de-inflammation? Based on what you're saying about inflammation and the cytokines and what we all kind of know about what goes on, is there something that we can do to de-inflammate and find the limit that we can de-inflammate and why it happens?
Well, certainly there have been suggestions that, periodically taking medications that have an anti-inflammatory effect, like aspirin or ibuprofen, low doses will suppress this background inflammation, and that's been recommended in a number of studies.
Okay.
And increasingly more, it's the bacterial colonies in your gut or your microbiota, as I mentioned earlier, that seem to be the initial spark in that starts the whole powder keg going, So if we can find ways to reduce inflammation in the gut, promote a more diverse microbiome and more of the sort of positive sort of types of bacteria rather than the ones that favor inflammation, that can also be a strategy. So, you need to look into sort of foods or diet that doesn't sort of promote inflammation so much. I'm not a nutritionist, but I'm kind of guessing that junk food, lots of sugar, all the sort of stuff that we know we shouldn't eat is probably encouraging colonies of bacteria that are detrimental. You'd obviously need to do some more digging, but I understand things like whole grains and, plant matter and fibrous materials are generally better for the gut biome than McDonald's. As much as we like McDonald's,
there's certainly naturally occurring compounds like turmeric, for example, and other spices that are known to reduce inflammation. So certainly, there are lists of them out there in studies. Introducing those into your diet probably be a really smart.
If I could just jump in with something here, other than sort of just refined sugar, that is just a massive inflammatory sort of inducing molecule. it really depends on sort of your genetic makeup as well. So there's been extensive studies done in Israel, I can't remember the exact name of it now, where obviously your genetic makeup will determine a lot of your internal gut biota. So you can actually have tests done now to see how well, from a sort of genetic point of view, that you're actually going to digest and break down certain foods, and sort of depending on, you know, how that's going to impact your insulin levels and things like that. So yes, there are certain foods that, you know, you can go online and you can search sort of anti-inflammatory diets and things like that, or ketogenic diets or veganism or things like that. But the reality is that there will be no single thing that is right for everybody, because we are so genetically different. So at the moment, unfortunately, without knowing at a genetic level how your gut biome is going to react, it's very difficult to determine, you know, whether chips are going to make you, you know, full of inflammation, or actually whether you're all right with them.
And for our American viewers at home, that's fries, by the way. Fries. We're not chips,
sorry.
not Doritos. Yeah. We're talking about fries.
All right.
Well, I do hope so, Olive. I do hope that fries don't cause inflammation.
Yeah, they're not French fries. They're American fries. Right?
American fries, French fries.
Congress changed the nomenclature years ago. We don't refer to them as French fries anymore.
Before we start drifting into something which might start an international political incident, next.
Okay.
Can I just intervene here?
Mm-hmm.
It's not really a study on inflammation, but it's just a study where they looked at the composition of your microbiome and changing in your diet. And what you can see is that all people, in broad terms, fall into 3 enterotypes. And changing your diet really can shift you from one enterotype in another enterotype. So it does seem that diet does have a huge influence. Though, of course, yes, you are completely right that genetics also plays a role. And, of course, the enterotype model is also a very broad category. And, you know, it's a combination of many different bacteria, and your genetic background will determine the exact composition of bacteria you will find within that enterotype, as well as other factors like exercise have function in this. And so all of this, and of course, you know, the bacteria you're exposed to, especially in early life. I mean, if you are never exposed to a bacteria strain, then of course it cannot occur in your gut. And there is actually evidence that it might be that humans over the last few hundred years have actually lost some of the bacteria. So you can go to, populations in Africa and so on who still live traditionally, and you will find some of the bacteria in their gut that you can no longer find in European populations, for example. So apparently it's presumed that they were once here in our microbiome as well, but they were lost. And if you are never exposed to them, they can never occur again. So, but, you know, I think that diet does have a huge role to play in the exact composition of the microbiome.
Okay. So just to pick...
just real quick, that's what I was trying to get at, was it doesn't seem like there's been no standardization on how to, in a research setting, lower inflammation with exactly this amount of turmeric or this amount of this amount of a pill or this amount of a, you know, it seems like if we won't be able to figure out inflammation until we figure out de-inflammation in a way.
And it's interesting to note that in plant material and supplement wise, things like bioflavonoids and things like quercetin, which is a senolytic. You've got teostilbene, which is in blueberries. Again, that has antimicrobial, anti-inflammatory action. resveratrol, which is related to pterostilbene again, anti-inflammatory, antibacterial. A lot of these related bioflavones and flavonoids and flavanols, which are all found in natural fibrous plant materials, a lot of them are turning out to be either antimicrobial, anti-carcinogenic, or a combination of those, and some of them are senolytic. So it begs the question, does eating a mostly plant-based diet drive down inflammation? And I'm not a betting man, but I think the smart money would suggest that, yeah, it probably does.
So, kind of piggybacking on what Sven talked about regarding the microbiome and the connection with diet and genetics, there was a paper published from France. I believe it's looking at the source, École Polytechnique Fédérale de Lausanne, EPFL, where they looked at these compounds found in pomegranate called urolithins. And the impressive thing with these compounds is, I think according to the study, they're the only compounds that are known to promote mitophagy. And the interesting thing, so as if that's not interesting enough, the precursor compound needs to be changed into the active form by your gut microbiome. And not everybody has the right gut microbiome to do that. So the kind of the take-home message from the study was that pomegranate, or the urolithin that's found in pomegranates, may be of more benefit to certain people than other people. And this is attributed to the gut microbiome, which in turn may be attributed to the genetic differences between the individuals, or it may be an environmental difference. I'm not sure. But that's a rather, kind of complex string of events. So I believe the researchers are trying to create the active compound right now so everybody can take it, rather than hoping that you have the right microbiome, that pomegranates would be super helpful to you. Yeah.
But that's a bit similar to another thing that is negative, and that is that the gut microbiome also transforms phosphatidylcholine or another choline type of products and betaine into trimethylamine, which has been linked to an increased risk of heart disease and stroke. So, that's another example of how the microbiome can transform a molecule, either making it good or bad.
Yeah, and there was also a recent study in China that was concluded, a large clinical study, and they looked at the microbiomes of healthy young people and very healthy aged people, and we're talking people in their 90s, even 100. And what was very interesting was the microbiome makeup, the mixture, the community of bacteria in both sets of ages was essentially identical. So there was very, very little in the way of inflammation. And that's very significant, because it suggests that it really does play a big role in starting off inflammation. And these people were very healthy, in their 90s, even hundreds. So and they had a gut microbiome like somebody who was in their late 20s, early 30s. And yet somebody their age, somebody else equivalent to their age, had a shocking biome. So,
but was it correlation or causation? Because yes, they have the microbiome of a young person, but is it because they have the gut of a young person, so therefore they have better stem cells, and and then they're basically their gut can support a microbiome that is more heterogeneous, more robust, like a young person? Or is it both, or is it some of each, or is it one or the other? I don't know.
Well, that's what we need to find out, and that's why they're and I've got to mention it. I'd be remiss if I didn't. It's pretty disgusting, but they're now experimenting with what most people would call poop therapy.
Well, that's been going on for a while, yeah.
Yeah, where they're actually, but this is the basis of it. They take the microbiome from one person and they put it into another to hopefully emulate the effects of, the health benefits.
Well, this is already approved by the FDA for treating C. difficile. Yeah. It's basically the last resort treatment when antibiotics do no longer really help.
Should be the first resort. Sounds like.
Well, I mean, it's the last resort because they are still fearing for, our lack of understanding of the microbiome and the risk that you could actually induce other types of diseases because we take it from donors and we really don't know what happens if you put it in another person. Maybe you get them obese and heart disease, or you get some other problems. So, they are careful with it, but, it is the last resort currently.
I think, to be fair, it'd have to be, wouldn't it, really, Oliver?
What?
It is also a psychological thing from the point of view of a patient as well.
I guess.
I fancy that.
Okay, so I'm going to share my screen. And I guess we're almost at the end. And this is, forgive me if I skipped over anything, but Figure 6 actually talks about how each of these things we discussed tie into one another, and it's the functional interconnections between the hallmarks of aging. And this figure right here, what it seeks to demonstrate is that it categorizes them as to what happens first and what happens next. Although the authors themselves admit that there's a lot of overlap, so it's not a strict chronology to what you see here, but primary hallmarks would be the ones that are basically kind of the rock bottom cause of damage, right? Genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis. So these are kind of really fundamental things that can go wrong. Although, I would say the epigenetic alterations, maybe that's due to loss of proteostasis or the other way around. So there's a little bit of, I guess maybe question as to, what's really fundamental. And then you have antagonistic hallmarks, which is the responses to damage. And this is where we get into these paradoxes that Steve mentioned. Deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, where a little bit could be a good thing. It could be a body's response to the primary hallmarks of aging or the damage that accumulates. But eventually there comes a tipping point where this all leads into the integrative hallmarks, which is kind of the which really is the manifestation of all of this, the phenotypes of aging, if you will, which is stem cell exhaustion, running out of stem cells, and altered intercellular communication, which basically ties into cellular senescence and the senescence-associated secretory phenotype, and basically all these misregulated cues that you get that cause inflammation to accumulate. And then it becomes a positive feedback loop, which ends pretty badly. Is that a decent summary? So I guess the question is so one question I had after I finished reading this paper is, if we go back to all those groupings, the way all of this is categorized, there is a lot of overlap. There's some exclusivity between each of these, and the question is, can we get a more robust response if we have a therapy that targets multiple points, right? So we're not just talking about, taking metformin or taking just rapamycin, or, upregulating telomerase, but doing a number of things all at the same time if they target kind of different areas of each of these categories. So if this is all correct, then we should see an additive effect or a synergistic effect if we have therapies that target each of these distinct, I don't know how distinct each of these are, there is overlap, but it would be worthwhile to see if targeting excuse me, targeting ones that we appear to be separate, if that has an additive effect, or if it doesn't have an additive effect, maybe they are somehow too interrelated. Maybe that's would be one way to tease apart if there is significant or non-significant crosstalk between each of these. Certainly there is, but I guess my bottom line is, can they be targeted independently, and can you have an additive effect if you do so with a number of therapies at the same time?
Yes.
Yes.
Yes, I'm going to say yes.
Okay.
I'm also going to say yes.
No, I think you're right. I think that there is going to be a certain amount of potential crossover, because all these hallmarks, well, they're closely linked, aren't they? I mean, one of the commonest links again is inflammation. It influences loads of other hallmarks. You know, a lot of people now call it inflammaging, which is a thing that's doing the rounds now, and I think it's an apt description. So yeah, I honestly think, for instance, if you targeted inflammation, various sources like senescent cells, maybe try and sort the gut microbiome out a little bit more, and if you did a few of those things that tamper down inflammation, you might see a number of different effects, like, for instance, stem cells and their regenerative ability. You're going to see a reduction in systemic inflammation in general, and you know it's going to have a knock-on effect potentially on various hallmarks. So yeah, I definitely think the answer to that question is yes.
But I would say pending further studies. Is anybody doing multiple approaches at the same time? A study which would target specifically 2 or more hallmarks using 2 or more different therapies at the same time?
Not at the moment, because I mean, really, what they're trying to do at the moment is perfect the toolkit and get everything in place before moving on to combinational testing. Because obviously, testing combinations is an absolute nightmare. And I mean, we certainly found this to be the case with mice, because in MMTP we were proposing to test multiple compounds, 2, even 3 compounds in a single mouse. It's a nightmare, because you've got to sort of, how do you work out what's doing what? So I suppose the same goes for developing, more direct rejuvenation technologies. They need to make sure that each one in isolation works as it needs to work before they then start adding them together. I'm not aware of any combination as yet. No, I think it will come. But I don't think it will come until they hone the toolkit properly.
Right. They certainly do combinations with cancers. There's been mentioned in the paper where you have, I guess, adding in metformin or rapamycin, something that's somewhat of a calorie restriction mimetic, along with whatever traditional therapy they're taking, is much better than either alone, right, in combination. So I guess, my point is that we might need to have a combination of 10 different things. And, can we do this? I mean, if we must, right? Maybe certain combinations will work even better than single approaches, right? Or maybe a single approach won't work at all. It needs to be in conjunction with something else to give you a much more statistically robust response.
Well, I think the logical one really to try as a kickoff would be stem cell replacement and senolytics together. And I say this because Alexandra mentioned it a few times, saying that it would be where she would go. And that's consistent also with what I mentioned earlier about the reduction of inflammation helps promote stem cell survival for transplants. So yeah, I can honestly see that being probably the very likely first combo, because they're already starting to do stem cell transplants with compound-based reduction of inflammation. So it's not really going to be a great stretch to do, say, dasatinib, quercetin plus a stem cell transplant to see how it facilitates. Definitely remembering that the SASP contains loads of different factors that inhibit stem cells. You know, TGF beta, IL-6, TNF alpha, and of course then our old friend NFKB. NFKB is always around, and I've heard people say, "Oh my God, NFKB, it causes aging." No, it's just a master regulator. But it doesn't mean it causes aging. It just means it gets out of control.
As many other things do.
It does, yeah. It's complicated, but I reckon there you go. That's my prediction. I think it's going to be stem cells plus senolytics. They'll try that as the first combo.
I'm thinking there'll also be combinations of small molecules, ingestible small molecules. You know, I mentioned the urolithin compound, which promotes mitophagy, maybe in conjunction with metformin, maybe in conjunction with, I don't know, some compound that prevents inflammation or slows down inflammation, such as ibuprofen or slow doses of aspirin. This term talked about a long time ago, you know, called polypill, right? Which you would have different compounds, and each of them would be validated for a different target, and together they would have either an additive or a greater than additive effect. Yeah, I remember when I was in David's lab, that was a term that I heard. But
I think ultimately, though, if you're going to have a polypill or something like that, it is still going to have to be almost genetically tailored to you, because like you were saying with the pomegranate extract, not everyone can utilize it. Right. So even if we did have this one pill hitting everything, depending on how we actually process all of those little molecules, you know, it might not work for some people.
Well, it could be certainly you know, the kind of the good thing about all of these molecules, or most of them that I mentioned, is that they're all generally recognized as safe, you know, given whatever dosage you're on. So all of these things, nobody's seen any significant harmful effects through chronic, you know, dosage. Metformin certainly is insanely safe, right? It's like you can eat grams of the stuff. People are actually better on it than not on it. You know, it's debatable whether or not this metformin should be in our water supply, but you know, and certainly for a lot of these other compounds as well. So, you know, the one thing I like about these compounds is that, you know, if they don't work, fine, they don't work. You don't get much of an effect, but they're not going to. Most likely, they're not going to damage you in any way. And if they do work, they'll work very well. So it's almost like a low-hanging fruit in that it might not work for some people, but it might work spectacularly well for other people. You know, some people might have genetically, they might already be optimized, right? People that are supercentenarians probably, or have the genes to be supercentenarians, certainly would not have to take a polypill, right? But the rest of us probably do, along with other things. All right. How's that for a wrap up? Are there any questions from anywhere?
What can we do about these hallmarks? You promised us solutions.
We just gave some solutions, didn't we? We gave a whole bunch of solutions.
Oh, you didn't tell me about DNA. How are we going to repair DNA?
We're just going to get rid of the damaged cells. We're just going to bypass the whole repair part. We're not going to use CRISPR, that's for sure.
No, there's someone was asking about this on the Discord channel earlier, funny enough. And there's two real approaches, aren't there? There's one, sidestep the complexity and just replace, which is, you know, replace stem cells, because you want to replace the stem cells because that's what's giving rise to all the other cells. And if, you know, that's how you get rid of damaged genome, is by replacing it from the top down. Or you can try and use CRISPR or such like to try and reconstruct a damaged genome. it's the more elegant solution ultimately, but I think we're some ways away from doing that yet. We may get to the point where we can do that, and I know people like George Church are talking about building genomes from scratch. And let's face it, if you can build a genome, you can certainly repair a damaged one back to a baseline. But I think we're going to be looking at replace rather than repair in DNA terms. And then you've got things like the Sinclair work, which is with NAD, which can also facilitate DNA repair. But there are certain fundamental kinds of DNA damage that just cannot be repaired, and that's where obviously replacement comes in. But if we're very, very lucky, Aubrey's going to be right, fingers crossed, and DNA damage will turn out not to be significant within the current human lifetime, rather than, as Hallmarks suggests, that it is. I'm rather hoping Aubrey's right about this, actually. But the thing is, either way, we need to develop the tools, either way, because if we're going to hope to live beyond 120 in good health, we're still going to need to do something about it eventually.
Okay, my question to everybody on this panel is anybody doing something about it to themselves? Yes.
I'm on a whole host of supplements and medications. I'm on the low-dose metformin. I'm on aspirin, please excuse my cat. I'm also on quercetin. I also take a number of plant alkaloids to try and, I'm trying to essentially reshape my gut biome after having a really nasty flare-up of disease after I contracted viral meningitis. It just completely destroyed my immune system. So I'm currently self-experimenting in as controlled an environment as I can get at home. Something's working because I am getting better and I haven't been well for about 2 years. And there are some symptom changes which I won't discuss here because they're rather disgusting. But yes, I do stuff at home. Whether or not in the long run it will have any lasting effect, we will just have to wait and see. But I am only 28, so there's a lot of time to see if anything works.
I want to know what we forgot to do as well, Oliver.
Yeah.
We forgot the extra missing hallmark, which we're tentatively going to suggest is a hallmark that wasn't included in the original.
Right.
It's very presumptuous of this, isn't it?
Yes, it is. So what is that hallmark, Steve?
Well, it's cross-links.
Yes.
Or some people call them AGEs, advanced glycation end products.
Mm-hmm.
But I just call them cross-links. But what are they?
Well, not all AGEs are cross-links, Steve.
No. There's always one, isn't there? There's always one. All right. Not all of them.
So, well, you have two categories, I will say. And you have those where a single amino acid residue gets modified. And surprisingly enough, there is no real good name for them. You could call them adducts, glycation adducts. And then the other category are the crosslinks, where two amino acid residues get like cross-linked together. So you have one amino acid residue, another one, and a cross-link forms between the two. Very likely, these two categories will have different effects. For example, the AGEs are known to cause inflammation. We are back to inflammation. And they do that by binding to cell surface receptors. Nobody has really studied this, but it's highly likely that crosslinks will maybe not do that. Because again, crosslinks will form between two amino acid residues, and so they are more likely not to stick out of the surface of a protein, and hence not be able to bind on a receptor very easily. While an H adduct, which sticks out, is more likely to be able to interact with the receptor. That's speculation, as I don't know anyone who has really tried to study that. At this moment, there is no way to specifically in a protein at least, in a peptide, yes, but not in a protein, to specifically build a specific advanced glycation end product in that protein, which is the type of technology that labs are currently developing and which will be necessary to really start to understand what they are doing.
Now, are these adducts and crosslinks, are they primarily localized, or not localized, they're all over the place, but are they found primarily in extracellular matrix and the exterior of cell surfaces?
They are formed everywhere, but most of the proteins inside your cell have a half-life of minutes to hours, with exceptions. One exception might be the nuclear pore complexes in neurons. So there was one study in Science a few years ago, like 2010, I think even, or something like that, where they looked at nuclear pore complexes in mice or rats, and they found that after a whole year, there was basically no turnover. In the liver, on the other hand, the nuclear pore complexes had a turnover, but not in the brain. So, okay, there might be some targets in the cell where advanced glycation end products accumulate, but the majority of the problem seems to be in the extracellular matrix, because there proteins have there are many more proteins that have a very long half-life time, some of them decades long, like certain types of collagen in tendons and so can have extremely long half-lives. And over that time, advanced glycation end products accumulate. And, the sad thing is that when advanced glycation end products accumulate and crosslinks accumulate, the collagen or whatever molecule becomes even more resistant against degradation. And so it accelerates its own accumulation.
Yeah, and the interesting thing to note as well is AGEs are more sort of prevalent in people with diabetes, and they contribute to things like diabetic neuropathy. So not only is it interesting from an aging point of view, I sometimes think that some of the symptoms of diabetes are kind of like accelerated aging in a way, from a sort of AGE point of view, and to go back to deregulated nutrient sensing, they kind of are.
And stem cell depletion.
Yeah. So kind of, my friend's a type 1 diabetic, and I was saying this to him, and he said, actually, that's probably one of the best descriptions of the disease I've ever heard. He said, because it is kind of like that. So yeah, it is accelerated hallmarks really in diabetes. So if we can actually develop ways to remove or cleave or break these crosslinks, we could potentially use it to treat things like diabetes. Hypertension is another classic for AGEs, because as these webs of tangled fused sugars, they become, very, very rigid, and like your skin sort of is very elastic, it stops being elastic. So as your blood's pumping through the system, normally it moves and sort of cushions the force, but more AGEs mean it doesn't, it resists it. So it's literally like a hammering action. And the problem is, instead of the arteries and things taking the force of the blood pumping around, your organs take the hit directly, and that's the problem. So if we could actually cleave crosslinks and remove them, we could actually do a lot for people with hypertension, which a lot of people get as they get older. So I think it's really important. And the reason I think it wasn't included in the Hallmarks originally is they just didn't know enough about it back then. I mean, some people say it's kind of incorporated into loss of proteostasis, sort of, but I think it really warrants its own category personally.
Well, especially because loss of proteostasis is really intracellular.
Yeah.
As generally, the extracellular matrix does not have any specific turnover rather than, enzymes secreted by cells and macrophages taking up the chunk. But, Yeah.
The one aspect that they mentioned was extracellular is aggregation, right? So that's certainly something that's correlated with, or positive. I believe there's a strong link between neurofibrillary tangles, which happen inside neurons, and then you have beta amyloid plaques, which happen externally, which are aggregates of normal proteins, and that basically end up being a sticky clump that, just like these ages, they can't be cleared out. They're accumulate, and they have potentially toxic effects.
Well, the order's yet to be ascertained, but I saw a study not long ago which suggests that it's beta that causes inflammation, and then that in the neurons, and then that causes beads to form, and then that's why you end up with tau tangles, which tau, if I remember, in a normal neuron, it actually transports. It's a signaling molecule that transports stuff up and down the neuron, but it gets dysfunctional due to inflammation from beta, and then causes tangles, and it just goes. That's what happens.
It's a MAP. I think that's microtubule associated protein.
It stabilizes the microtubules, and when it aggregates itself and forms these neurofibrillary tangles, then the cytoskeleton actually starts to fall apart. It becomes unstable, and that causes dysfunction in organelle transport.
Mm-hmm.
So anyway, well, on a positive note, with the AGEs, last year, as you may recall, the problem was doing something with glucosapine, which is the most common form of AGE by an order of magnitude. The problem was they couldn't actually reliably create it in order to test interventions against it. But last year, SENS Research and the Spiegel lab at Yale managed to synthesize it, and they can now produce it en masse very cheaply, which is great. But a couple of months back, or a month ago, SENS announced a new project, and they're actually now working on raising antibodies against glucosapine, particularly monoclonal antibodies. And I understand that they've actually got some candidate enzymes as well. So fingers crossed that we can get some results from that soon, because that has huge implications for aging and a variety of diseases. So, go SENS Research Foundation.
Yeah. So so if you put an antibody on glucosapine, it's going to attract macrophages. The macrophages are going to eat it, but are they going to be able to digest it?
Well, that's what the enzymes are for.
What, you mean modify like a macrophage to?
I would think so. Yeah. I mean, obviously Sven worked on that side of things, but I would imagine that's what they're going for, is to give the macrophages the tools that they need to break this stuff down. Because I definitely know that they've got some candidate enzymes. So
very cool.
It is very cool. I'm exceedingly excited about it. I can't wait to see some results, because we know that there are breakers. There must be breakers, because we know that there are breakers for other kinds of AGEs, right? Like carbonyl AGEs. They can be broken down by rosmaric acid, for example. So we know that these cross-links, other kinds of cross-links and AGEs form, and there are things that do definitely break them down. So it's just a case of we've got to find out what it is for glucosapine in particular and deal with it. I think we'll get there.
Well, certainly the Hallmarks of Aging paper is I think, a nice beginning of a roadmap for for tackling kind of the central, the central causative points of aging, at least mammalian aging, human aging. I'm also interested in kind of, I see they're in the aging or longevity field. There's like a dichotomy between two groups. One is, the biomedical community, which is interested in, human lifespan and a lot of this paper, most of this paper here pertains to that. You know, it talks about mammalian aging and of course model organisms that are invertebrates, but all again plugging into mammalian aging, human aging specifically, and what we can do to tackle it. And then there's the kind of broader picture of aging and longevity mechanisms, which is universal, right? Which is the evolutionary biology of aging, or rather the evolutionary biology of longevity mechanisms. And there being some a lot of scientists, including Leonard Hayflick, who would, posit that, we know what aging is. It's basically a manifestation of the second law of thermodynamics, things breaking down, and systems had to evolve longevity mechanisms to thwart this from the very beginning, right? And it's always been there. And we certainly see a lot more evidence these days that it is a global phenomena, depending on how you look at it. Yes, there's certain cells certain organisms that seem immortal, like Hydra, but you have to keep in mind Hydra also has a constant cell turnover as an organism. It's also there's no real distinction between germline and somatic cells. The cells, yep, as I'm aware, they can basically differentiate, if that is a good term to use, between one or the other. So different systems have probably evolved different longevity mechanisms to deal with the damage. We've seen now, for a long time, people thought bacteria don't age, but they actually lose their replicative potential if you track individual bacteria. It hasn't been done with every bacteria, but certainly with E. coli and a couple of others. So it could be that, what we're really looking at is the evolution of longevity mechanisms, and there could be lots of overlap, but there could be, there could be there could be differences depending on the system. And for humans, the mechanism by which humans have evolved to deal with aging fundamentally is, I think I mentioned this before, is that we shunt all the garbage to our somatic cells, and, we have a pristine germline that keeps going, right? And the problem we have is that we've grown attached to our somatic cells because our somatic cells have now grown a sentience, and they are now self-aware, and they don't want to be cast off.
I blame the mitochondria personally. Those.
You think it all went downhill during endosymbiosis 2.5 billion years ago? That's when it all started to go downhill.
I think so. Those parasitic little stowaways have got a lot to answer for, because as wonderful as they are at generating lots of energy for the cells, that energy comes at a price, of course, and that is a lot of stuff can go wrong with them. So I think we should get rid of them. Never mind about transferring the genes to the nucleus, as SENS is proposing to do to reduce the ROS damage. One solution, by the way. I think we should just get rid of them and re-engineer the cells to produce energy themselves. I mean, look at the cancer cell in the with the Warburg effect. They essentially make mitochondria become dormant or a very low energy generating state, and yet they generate vast amount of energy. So let's steal that mechanism.
Are you proposing to become a tumor?
No. We obviously want the energy generating pluses without the turning into a giant ball of cancer.
And that's essentially what one of our first projects in LEAF was. It was funding a SENS project where they were trying to, I guess in their words, accelerate evolution and stick the rest of the remaining genes that are found on the chromosome found in mitochondria. I think there's 13 of them back into the nucleus, and thereby protect the genome. Of course, that's not doing away with the mitochondria at all,
no,
but it is buffering its genome against the damage which is happening in mitochondria.
And the interesting case in point to mention there is there are already about 1,000 genes, mitochondrial genes there already in the nucleus, because nature itself has been slowly over millennia moving them from outside the nucleus to the nucleus. So it's not like we've suddenly gone, hey, we've got this great idea, No, nature's already had that idea. We're just accelerating it, as you say, and it does show that on a significant level that slowly but surely nature is selecting for longevity. It is.
Mm-hmm.
And it's doing it in other ways as well. But mitochondria and its migration to the nucleus, the genes, is probably one of the best examples I can think of where nature is actively selecting over a course of time for longevity.
Well, it depends. Yeah.
Evolution, tries to set for each species the optimal lifespan, and the optimal lifespan is that the animal lives long enough as it will be expected to be able to survive in its environment. Its environment depending on the predators and the environmental conditions like food availability and all of those factors. So if an animal is expected to live for 50 years in its environment, then evolution does not build a system so robust that the animal can live for 1,000 years, because that's just wasted energy.
Well,
yeah.
I was just going to say, just to comment on your comment there, that evolution by natural selection. I mean, the evolution doesn't really care so much about individuals, but rather the maintenance and the success of populations, right? So yes, natural selection works on individuals, but the population evolves over time. And as long as the population keeps propagating, it's a success. It doesn't matter if the individuals drop dead, right? So, okay, mice have a 3-year lifespan, 2-year lifespan, but they've been around for how many millions of years, right? As a species. So that's, as far as evolution is concerned, that's successful, right? Ants, I don't know what their lifespan is, but it's probably pretty short. But all the different species of ants have been around for also, for eons of time. So the individual lifespans don't really play much of they do play a role in the sense that given a particular environment at which the organism is evolved, you need to have, you need to compete with other organisms and produce sufficient offspring to keep the population growing. And in one environment that may need or may, the selection may push it towards a 50-year lifespan, another, perhaps a 2-week lifespan. But again, evolution is only concerned with maintaining, the population or the species, and if that's not maintained, then it goes extinct. So it's sort of, in that respect, lifespan is irrelevant, to evolution. It evolved as a consequence of the environment that the organism was in, and yeah, we could talk about antagonistic pleiotropy and the ways that these longevity systems evolved, but I think fundamentally you do need some longevity maintenance systems, and that these are very ancient. For example, DNA repair. Everything has to do DNA repair, whether it's a prokaryote, archaebacteria, eubacteria, whatever, humans. But the efficiency is going to be dependent on the particular environmental niche and the selective pressure that the organism is in.
I agree, but I will just frame it a little bit different. I will frame it in the sense that within a certain environment in which an animal lives, puts a limit on its lifespan. An ant cannot live 50 years because the predation risk and things like that is too big. So that limit exists there by the environment, and what natural selection then does is ensuring, during evolution, that the animal has the lifespan, that it lives long enough by evolving these protective mechanisms to live that expected time it can remain alive in that environment. And so, a human who can live longer, evolution has evolved us to be able to live that long. You see what I mean?
Sure. There's other things that, you know, evolution is going to come up with tricks in the bag to maintain the species, which is, you know, short lifespan, many offspring, long lifespan, few offspring, right? So it's all about the maintenance of the species, the competitive fitness, if you will, of the species.
It's the ants I feel sorry for. Although there is actually a species of ant that's negligibly senescent. They found out, I think it was last year or the year before. There is one type of ant that actually is.
Well, the queens live a long time.
Yeah, the actual workers and everything, they're negligibly senescent, so they don't have physical manifestations of aging. I don't know why that developed in this particular species of ant. I don't know.
I would be very interested in looking at systems that are considered, you know, non-senescing or non-aging, and see, because, you know, my argument would be that the individual cells of the ant are accumulating damage, right? There's certainly no, if a species has been around for 2 million years, there's no 2 million year old cell anywhere to be found, right? It's just the information pattern that's recycled. So clearly there must be some sort of damage accumulating at the cellular level, and the ant has to be
good at repairing it,
doing something to deal with that.
The first thing I would look at, and this is definitely my first port of call, any very, very long-lived species or negligibly senescent species, I would always look at how stable the environment is, because in general, the longest-lived species inhibit extremely stable environmental niches. So things like the Greenland shark, okay, that lives for what? We think 800 years plus, okay, which is shocking, right? You know, some of those sharks, they reckon, were swimming around before America existed. I mean, it's a kind of crazy thought, right? But what's significant about their environment is it's super stable. You know, think about it, large bodies of water, the temperature doesn't change very fast. It's very stable. You know, the environment is extremely stable, so they live longer. And the same as is it the sequoia?
Yeah.
Sequoia that lives, I think that's one of the longest lived trees. Again, it inhabits a very stable environmental niche. And that huge African tree, is it the baobab?
Something like
it's something like that.
Baobab.
There's one comment I want to make about plants. I mean, it's a little different with trees in that, again, the giant sequoia tree, you know, and there's some other trees, I forget what they're called. It's like some sort of creosote bush or something, which has been around for like almost 1,000 years. Again, none of those cells are 1,000 years old. There's a turnover of these cells. And with trees, especially with plants, somebody once summarized it to me as saying, you know, pretty much every cell in a plant is a stem cell, right? Meaning that there's high plasticity that's being exhibited there. So, you know, in one sense, you can almost see certain organisms as, I don't want to say a population, but, you know, it's the fact that their germline and their somatic cells are not very clearly differentiated means that they, you know, might have a mechanism in place that's getting rid of cells that are damaged in that wouldn't be really possible to implement in an organism that has a sharp distinction between germline and somatic cells. And that the strategy that those organisms evolve that have a sharp distinction between somatic and germline may be that the somatic system is being just jettisoned, right, for the benefit of the germline. But obviously you can't jettison that in an organism that has no distinction between a somatic or germline. Otherwise you jettison everything and the organism is dead and extinction happens. So there's probably a constant turnover of every cell, in a giant sequoia, a hydra, that yes, the organism itself, is 1,000 years old, but I think I want to just emphasize to people listening that I doubt very much, and I don't think anybody states this, that there's any single individual cell in a hydra or in a sequoia that's 1,000 years old.
But negligible senescence does also not automatically mean long-lived, because I think your N species is probably quite short-lived from a human perspective. It's maybe a few weeks, a few months, but it's not going to be years. And what negligible senescence means is basically the risk of dying does not increase with age, right? But if the rate of dying is 10% per month, then after a year all these animals are dead. They are negligible senescent, but they are still dead after less than a year.
So why didn't aging? So why are there? So wouldn't we predict in all these models that the ants should rapidly age if the antagonistic pleiotropy model is correct and all the other models are correct? Shouldn't the ants reproduce and then start to age? Why would they have a system that's keeping them going?
Maybe evolution selected for them to be very, very good at raiding your picnic basket. I don't know. I'm sure these answers will come to light, and in a jam sandwich near you.
Yeah, I have a feeling that a lot of these, aging, and longevity, both go hand in hand. You have mechanisms that try to thwart aging. There's a lot of paradoxes in this entire field, probably more so in every other field. But I think, that's a good thing, because I think these paradoxes will be resolved. They will be resolved, and there will be an explanation that basically resolves them, and that we might see a unifying kind of theory of longevity mechanisms that can be applied across the board, and with differences, different strategies being employed by different organisms, metazoans versus unicellular organisms.
So basically, you want a unified theory of aging. Well, Hallmarks is about as close as you're going to get to that at the moment. Yeah. But we're obviously only looking at mammalian species with a particular emphasis on humans, because, well, we are.
Yep.
And I don't know about you, but I'm not that excited about roundworms. Although I have to laugh whenever there's a new experiment that extends the life of C. elegans or some such. New supplement, new compound found in, I don't know, strawberries increases the life of C. elegans by 10 times. Everyone's like, wow, everyone's rushing out. I'm like, guys, you do realise C. elegans lives for like a few months. It's really not really that important. But there we go. Always makes me smile.
All right. With that, happy Halloween, everybody. Do we have anything to add before we sign off?
And what a horror story and a Halloween horror story we've told today. Aging, probably the most scary story ever, right?
Right. We are our own worst enemy.
Well, yeah, this is it.
Our cells turning against us.
We will do something about it.
Maybe a quick Halloween joke. Senescent cells or zombie cells. Yeah. That's also the title of a Nature, news story last week. Because there are cells that keep living, but they do not die. They just keep living in this, metabolic inactive way. So kill the zombie cells.
That's it. Senolytics is the key. Never mind about Van Helsing. We've got Senolytics.
That was vampires.
Didn't he kill zombies and stuff as well? I thought he was a sort of. I thought he was a generalist. He was a Renaissance man. And he went after werewolves as well in that one movie. So he did a bit of everything, really. He was your odd job man, Got a ghoul or a vampire, a ghost? Call Van Helsing. 0800 Helsing. I don't know.
Right. I think we're wrapping it up for today. And do we have any paper that we're going to discuss a month from now? One paper that I was interested in, not that we're going to discuss this next month, is the paper by Li Wei Sai, dealing with 40 hertz stimulation, light stimulation, and its effect on an Alzheimer's mouse model, and how that potentially can ameliorate or reverse Alzheimer's symptoms. The reason why I think it's interesting is, number one, 40 hertz light treatment is pretty easy to do. And number two, if you go online right now, people are selling kits. People are trying to self-experiment with this. So it might be important to talk about what this is all based on, right? It's based on this one paper. If there's other papers, maybe, but I think it's primarily Li Wei Sai's paper. So we're going to toss that one out there, and it's pretty recent. And I think what 40 hertz stimulation is supposed to do, it's supposed to promote macrophage clearance of beta amyloid plaques, although don't quote me on that, I may be totally wrong.
That's interesting. Well, I mean, everyone's free to let us know. And I mean, everybody, not just everyone here. I mean, anyone listening is free to suggest some ideas, what you might like us to cover. In fact, we've done a few of the suggestions that people have sent in, and they've been quite interesting. I mean, otherwise we'll just end up talking about what we want to talk about, which,
if I may interject here, that's part of what myself and Dr. study is traumatic brain injury, post-traumatic stress disorder. And I led into this because I wanted to know why I didn't commit suicide. We weren't sure what and or how, and my memory got so bad I couldn't remember my combination, my cell phone number. I couldn't remember hot or cold in the shower. So I swore to God I had to mention it. I knew it. I was wrong. The plaque and the beta amyloid plaque. I started off in a procedure with what's called brain beam. It's through a Dr. Nelson here in Reno, Nevada. He's kind of a freak in a way. His mom died of Alzheimer's, and he's a functional medicine chiropractor who's known people in the supplement industry for decades. And he's come up in the ranks. And I believe the Russian gentleman who created the tonic in Russia to counter our methamphetamines, Dr. Nelson took over that patent from the gentleman who was brought over here from Russia. And it's called Provite. It's a super adaptogenic tonic. It's one of the things I'm using. But he also developed Brain Beam. It's I believe each teaspoon is 3,000 milligrams of C3 sabenza curcumin, standardized, with Ceylon cinnamon, uracemium, and cordyceps. And I swear it was as if the ADD was lifted. I used to always say, what was I saying? What was I saying? What was I saying? Just constant. And it's like it's gone. Now, about a month or two later, I started Samoralin, which we call the Holy Grail here. And so I can't correlate if whether it was just the brain beam that did it. But he's also started in on brain immune and collagen peptides. Because I've been on that bulletproof coffee now for a couple of years because I might have that lipodystrophy. I don't produce fat. So I slam 4,500 calories, 5,000 calories a day with just, I mean, I've been eating here the whole time you guys are here. I eat nuts and seeds all day. I eat 2 things of cashew peanut butter a week, avocados. I mean, I just slam food, but I'm doing it and it's working. So I know there's ways to keep lupus, keep lipodystrophy, keep gulf war illness, keep all these sicknesses at bay in a real weird equilibrium that I'm figuring out and finding out. Now there's collagen, and then there's collagen protein, which comes from Bulletproof. Dr. Nelson came up with a collagen peptide that he believes is far more superior and less likely to be dairy. based off of eggs, I believe, and the coconut milk. And it has to do with stabilizing the gut. The same thing with
Joel, sorry to cut you off. Is there a link that we can mention to people that they can check this out?
Right now, it's probably just brainbean.com.
Brain what?
Brain Bean, B-E-A. Brain.
B-E-A bean, and like a jelly bean.
Yes, correct.
Brainbean.com. Say that 3 times fast.
Best
all right.
bet. And I'll try to get a name and an email and all that stuff and get that to you, because he might be onto something that we need to get out there, just to let you know. Because I just got word just now. So Jaden, my nephew had an abscess on his elim. It might be connected to his colon. They say it's Crohn's. Your typical medical system here in America, they want to give him Humira. We went into natural foods. We went into this Brain Immune. I did the Provite. And according to her, his abscess is basically gone. We knocked it out the park in 2 weeks, have MRIs to prove it. And so there really might be something to what this doctor is developing. And I'll keep you guys updated, study it as much as possible, get the information and the links to you.
Yeah, please do, because the modern medical establishment, of course, isn't really set up to deal with dietary interventions.
Correct.
Anyway, it is 2:56 right now. I have another meeting to go to in 4 minutes at 3 o'clock. So I think at this point, this is a good wrap up.
Yep.
Okay, everybody.
Okay.
If you're going to a Halloween party, stay safe. Don't take candy from strangers. Have a good night.
I always take candy from strangers. That's how I get candy. I mean, come on.
I'm the stranger that gives out the candy. That's me.
Oh, I'll be after your sweets then.
Thank you for what you all do. Seriously.
Thank you.
Thanks for joining us, and we'll see you again next time. Thanks to our patrons for making this possible. Thank you very much. Bye.
Bye,
See you next
everyone.
month.
See you.
Bye.
Bye.